Hi, this is Diana. Today we are going to talk about the Lysine agar. The Lysine agar is a differential agar slant, which is to test the ability of an organism to deaminate the lysine or decarboxylate the lysine. And the medium has an aerobic slant, which is exposed to the air, and an anaerobic bud, which is without the oxygen present. A brown crystal purple is a pH indicator. When the pH decreases to 5.2 or even lower than 5.2, the color turns to yellow. However, when the pH increases to 6.8 or even greater than 6.8, the color will become purple. The glucose is a component in this agar slant and can be fermented in the bud of this medium. And the acid and products of this glucose fermentation change the color of bud to yellow color, which we just mentioned here. When the pH decreases, the color will turn to yellow. The lysine deamination is an aerobic process which occurs on the slant of medium. Here I have been listing some major products of this lysine deamination reaction. If there is an organism that can produce the lysine deaminase, and the alpha-keto acid will be formed. Alpha-keto acid can react with the iron salt, which is a component of this agar slant on the surface of the medium. Then the red compound will be formed. We use capital letter R to represent the formation of the red compound, which indicates there is an organism that can produce the lysine deaminase. So R, which means the lysine deaminase positive. Next, we are going to talk about the lysine decarboxylation reaction. The lysine decarboxylation is an anaerobic process which occurs in the bud of the medium. If there is an organism that can produce the lysine decarboxylase, the cadaverine will be formed here. The cadaverine is a phosphoryl diamine compound produced by the animal tissue. The cadaverine can neutralize the acid, which is formed by the glucose fermentation. Then the alkaline state will be formed here, and it's formed in the bud of this agar slant. So we use capital letter K to represent the formation of the alkaline state, which indicates there is an organism that can produce the lysine decarboxylase. So K means lysine decarboxylase positive. And A here, which represents the acid. If the bud of the media is acidic, which means there's no cadaverine formed, so that there is no organism that can produce the lysine decarboxylase. So A indicates lysine decarboxylase negative. Let's take a look at the test result marked on the tubes here. There are two letters here, and between them there is one slash. So the first letter, which is on the left side of the slash, stands for the result of the lysine deamination, which occurs on the slant. And the second letter, which is on the right side of the slash, which stands for the result of the lysine decarboxylation reaction, which occurs in the bud of this medium. For example, let's take a look at the test result on the first tube. So first K here, it represents the lysine deaminase negative. And the second K here, which represent the lysine decarboxylase, positive. Here I want to mention the hydrogen sulfide here, H2S. Like we talked in the previous video of hectonenteric agar, Salmonella species is an organism that can reduce the sodium thiosulfate, which is a component of this medium, to hydrogen sulfide, H2S. And then the ferric ammonia citrate, a source of the iron, which is another component in this medium, will react with the hydrogen sulfide here. And then ferrous sulfate will be formed, which is a black precipitate in the butt of the tube. So you can see here in the butt of this medium, it's pretty dark here. So which means that there is a ferrous sulfate formed, which is a black precipitate in the butt of the medium. So in this Lysine agar test, you need to know how to interpret the test results. And thank you for watching.